Hello, everybody, and welcome to our webinar on SCORE and ClinEpiDB. We will give participants 2 more minutes to arrive and then get started. Hello, everybody. Thank you for joining us today as we talk about SCORE and ClinEpiDB, a demonstration of NTD and schistosomiasis, data accessibility and exploration. My name is Nupur Kittur. I'm a data specialist with ClinEpiDB. Joining me today to host this webinar are my colleagues, Danica Helb, Steph Schulman, and Sheena Tomko. I have a few housekeeping notes before we begin. All participants are automatically muted, but we do welcome questions at any time. Please type in your question in the webinar control panel right here. And this presentation is being recorded and will be emailed to all participants later this week. ClinEpiDB is part of a larger bioinformatics project called ViewPathDB, and we would like to acknowledge the support of the ViewPathDB team. Also, we would like to thank all of our funders. In the next hour, we will learn about the purpose of ClinEpiDB and get an overview of schistosomiasis datasets on ClinEpi. This will include a brief introduction of schistosomiasis and the SCORE project, followed by a live demo exploring one of these datasets. What is ClinEpiDB.org? ClinEpi is an open-access clinical epidemiological database resource encouraging online exploration of complex studies. The paper referenced here will give you a lot of background on this project. The purpose of ClinEpi is to make data fair, that is, findable, accessible, interoperable, and reusable. We make data findable with unique study identifiers. We allow users to access data in various ways with a search wizard that allows complex searches and analysis apps for visualizing data. We make data interoperable by mapping variables to harmonize terms across datasets. And we provide study pages that explain the context and the methods of a study. There are currently 26 datasets available on ClinEpiDB, with new studies being added every couple of months. These are data from studies that focus on various diseases such as malaria, schistosomiasis, enteric, and respiratory diseases. Various types of studies are included in ClinEpi, We have observational data from case control, cohort, and cross-sectional studies, and experimental studies, including randomized controlled trials and cluster RCTs. The data you will find on our site is of diverse types that includes geographic data, socioeconomic metrics, and household characteristics, as well as individual level data on demographics, clinical data, sample assays, and much more. Recently, 9 schistosomiasis studies have been released on ClinEpi, and you can see them listed here. So let's start with a very brief introduction to schistosomiasis. Schistosomiasis is one of the 18 infectious diseases that are termed neglected tropical diseases. Neglected tropical diseases, including schisto, disproportionately affect the poor. They are largely treatable and preventable, and they are disablers rather than killers. Schisto affects over 200 million people in over 70 countries. But as this map shows, the vast burden of schisto is in sub-Saharan Africa, and that is where all 9 of these studies were conducted. Schisto is a parasitic disease caused by trematodes or blood flukes of the genus Schistosoma. Worm pairs live in human blood vessels of the intestine or the bladder, depending on the species, and these schistosomes are about a centimeter or 2 long. The classic way to diagnose schisto is by detecting and counting parasite eggs in stool or urine. Freshwater snails are intermediate hosts for schistosomes, and so schistosomiasis has a complex interplay of people and human water contact and host snails who live in rivers, ponds, lakes, and streams where the humans go. Schistosomes live for 3 to 10 years within their human hosts, eating erythrocytes and producing hundreds to thousands of eggs daily that end up in host urine or feces or get trapped in host tissue. It is the schisto eggs and not the worms themselves that are the problem. The 2 major schisto species in Africa are S. haematobium and S. mansoni. S. haematobium causes urogenital schistosomiasis, with complications that include bladder carcinomas and an increased risk of HIV infection. S. mansoni causes intestinal schistosomiasis, with abdominal pain and hepatosplenic fibrosis. Schisto also causes several non-specific disabling morbidities, such as chronic anemia and decreased physical fitness and cognitive function. Luckily, there is a good drug called praziquantel, which is effective against schisto. Praziquantel is a well-tolerated oral drug that kills or impairs schistosomes and drastically reduces egg output. And this drug is suitable for delivering on a population level by mass drug administration, or MDA. The SCORE project, or the Schistosomiasis Consortium for Operational Research and Evaluation, was a large multinational effort that worked on many aspects of controlling schistosomiasis. SCORE focused on MDA with praziquantel and on practical, real-life questions with a goal of better decision-making at the program and policy level. A lot of their findings can be seen in the special issue that's linked here. Of the 9 SCORE studies on ClinEpiDB, 2 studies focus on controlling S. mansoni, 4 focus on controlling S. haematobium, and the final 3 studies focus on diagnostics. The first data set I will talk about today is the largest SCORE data set that we have on ClinEpi, and it is actually a combined data set from 4 separate field trials. The question asked here was, what is the optimal MDA regimen to control schisto at a population level? In particular, these trials compared community-wide treatment to treatment that was given only in schools. And they compared treatment that was done every year with treatment that had some drug holidays mixed in. Drug holidays are years in which treatment was not given. Two of the studies in this data set were done in areas of Kenya and Tanzania that had very high prevalence of schisto, and these two studies were called the gaining control studies. The other two studies in the dataset were done in areas of Kenya and Côte d'Ivoire that had relatively moderate prevalence of schisto, and these were called the sustaining control studies. The study strategy was to choose villages within the study area and randomize them to different MDA regimens. There were 25 villages in each of 6 study arms, and they then tracked prevalence and intensity of schisto in 9 to 12-year-old children in the study villages over a period of 4 years to evaluate MDA effectiveness. The findings of these trials have been published, and they show that all tested regimens significantly reduce schisto burden, but that none of them lead to elimination of infection. Secondary analysis also shows that annual treatment is better than including drug holidays in the regimen. So with this background, I'm going to go to a live demo. Let's take a look at the website, which is ClinEpiDB.org. Right now, you will see that I'm not logged in. I'm using the site as a guest. The advantages of logging in are that you will get to save your searches and come back to them. And if you want to download certain data sets, you will have to register, which only takes a minute, and fill in a data access form. But other than that, you are free to explore the website as a guest. On the home page, you see a header, and here is a drop-down list of all the studies that have been released. You see a workspace that lets you see search strategies. And the About section has a lot of helpful links to things like frequently asked questions and tutorials and resources. Below on the page, as you scroll down, you will see study cards. So these study cards, there is one for each study. The study card will have the name of the study and a few key features, such as study design, location, and years in which the study was conducted. The study card will also have 3 kinds of links. So you can go straight from the study card to look at the study details and learn more about the study. Or you can download the data and work with it on your own. Or you can explore the data right here on our website. Let's go back to the header and click on this link for all studies, which takes us to this handy table. Here you will see a list of studies highlighting some features of each study. You can see the title of the study, the disease under investigation, the study type, whether it was observational or experimental, and the study design. You will also see the number of participants, the location of the study, the years in which the study was conducted, etc. If you're interested in data from certain countries or specific years, this table is a good place to scroll through and look. The study accessibility column here tells you how much access a user will get to the data. And this level of access is decided by the study providers. Some studies are designated as public, which means anyone is welcome to download data, whereas many are protected, which means that you are free to explore the data, but to download it, you will have to fill out a data request form that the study providers will be asked to approve. You can search the studies in this box. So I'm going to search for schistosomiasis and click on the SCORE S mansoni Cluster Randomized Trial so we can look at it in some detail. Clicking on the title takes you to the study page. And here you can get an overview of how the study was conducted. So this page again lists several quick facts about the study. It will link to major publications that came from this study, and it will give you a description of the study. So here in this brief summary and objectives, we will learn that villages were randomized to receive MDA with praziquantel, and that the major outcome was prevalence and intensity of schistosomiasis, And the goal of the study was to determine how different approaches, school-based versus community-based, have an effect on schisto prevalence. And they also tested timing of MDA. Either the drug was given every year or biennially, which is every other year, or by skipping 2 years after 2 years of treatment. Here you can see a map of the study locations. We are going to be looking at data from Côte d'Ivoire, Kenya, and Tanzania. As we scroll down in the methodology, we learned that there are two types of studies that are included in this data set. The sustaining control study was done in Côte d'Ivoire and Kenya in relatively low or moderate levels of schisto infection. And the gaining control studies were done in Tanzania and Kenya in villages that had high levels of schisto infection. So Kenya is a country that had both types of studies done in it. Scrolling down further, we can see the study design, and we see that the target population was 9 to 12-year-old children, although they did collect data on other age groups too. So with some background on this study, we can now go on and explore the data. So we can click on this person icon, and it brings us to the search wizard, which will allow us to explore the data right here on the website. You see right away that this study has 233,000 participants. The search wizard gives you an overview of all the data in the study, grouped into categories. You can examine the basic distribution of every variable in this study. The first thing we see here are study details, such as the study arm and the study type, whether it was the gaining type or the sustaining type. Next comes the geographic region. You can see right here that the three countries that are represented in this study, and that GPS data is available. Notice that this will tell you that there's no data missing for this particular variable of country. You will see here that we have a table of the distribution of villages in different countries, along with a little graphical representation. And this tells you right away, for instance, that half the villages were in Kenya. Exploring further, we can click on village and click expand all to see all the variables that were listed under village. We can see that data was collected over 5 years. And here, the village level information, we see some important data on MDA coverage. It's telling you how many kids were in the village and how many kids were treated, which shows you the percent of kids treated, and the same for the total population of the village. If we look at this variable, total population of village, you can see that for a continuous variable, the website shows you a histogram. You can see descriptive statistics listed, such as the mean, the maximum value, the mean, the median. This will be listed at the top. You can scroll down to adjust the bin width of the histogram. So you can maybe change it to 500 to change the look of the histogram, or maybe 100. So you can see here that most villages had a population of between, if you hover your cursor, you will see that most villages, which is 43, fell between 300 and 400 people. So most villages had this range of population, and you had all these outliers that you can see here. How about if we look at percent of total population treated in the village, which is a measure of MDA coverage? Let's adjust bin width to 5. So what do we see here? We see here that most villages had between 75% and 100% coverage. So the coverage was pretty good. You do see outliers. You do see that there was a village that had very low coverage. And if you're wondering how more than 100% of people can get treated, it's because in some case, the study teams have let us know that people came in from neighboring villages to get treated. That's how you have a few villages that had over 100% coverage. So these are some of the features that make ClinEpi a useful resource for exploring details of a data set and becoming familiar with variables and their distribution right here on the website by simply pointing and clicking. So let's continue over to the participant tab. And here we see basic demographic information such as sex, age, and age group. The sample tab, again, I'm going to expand also, we can see all the variables. It shows us that stool samples were collected and processed to detect schistosomes and hookworm, ascaris, and trichuris, which are soil-transmitted helminths. For the schistosomes, we have two types of data. We have the mean egg burden for each participant, as well as a binary variable that simply tells us for every participant whether they were schisto positive or negative. You can also subset this data with a search strategy. So if we go back to the study details, here are the 6 study arms. Each one is designated by a 4-letter code where the S stands for school-based, C stands for community-wide, and H stands for drug holiday, or a year in which there was no treatment. So let's choose arm 1. So this was the arm that had treatment given every single year. In the case of the moderate starting villages, it was school-based treatment every year. In the case of the gaining study with the high schisto prevalence, it was community-wide treatment every year. But nevertheless, there was treatment every year. So when I clicked on arm 1, you see that my participants were reduced from 233,000 down to 60,000 because we only chose the one arm. Let's go over to geographic region and then choose Kenya. Let's say for this query, we only want to look at participants from Kenya. Here, the number of participants is going down further because we are subsetting again. Finally, let's go over to participant and let's choose only the 9 to 12-year-olds because this was the target population of the study. If you forget what we filtered on, just click on the green filter icon and it will pop up a list of the parameters that you have chosen. Here we have the ones arm 1 got treatment every year, Kenya, and 9 to 12-year-olds. So what you can do now is view your subset of study participants that you chose. So here it will start showing you a list. At this point, you are allowed to download just this subset and work with it on your own if you wish to do that. Or we can go ahead and do some basic analysis right here on the website. So let me choose Analyze Results. We see 3 visualization apps here, and these will allow us to look at distributions such as histograms, or make contingency tables, or make data summaries such as box plots. I'm going to click on contingency tables. So over here, I'm going to choose schisto infection. So I'm going to say, let me choose whether the participant had any schisto infection. And these apps just take a few minutes to run as the app talks to the database. So hang in there. The app will work it will update in a few seconds. Over here on the dependent side, I'm going to keep this as it is. It's the selected participants, or it is the subset that we selected earlier in our search. Okay, it's here we will update it to at least once positive. So let's say schisto positive over on this side among our selected participants. The plot further allows us to stratify. So let's go ahead and stratify this by time point. And while the app does its thing, please use this time to type any questions into the chat as you think of them while we let the app work. Let me also use this time to say that ClinEpi is currently working on improving visualization apps. So we are going to have new, faster apps available in just a few months. So here I've chosen the study time point so that we can see how the proportion of Schisto-positive participants will change over time. And then we will further stratify the plot here. Since Kenya, if you remember, had two types of studies, the gaining study and the sustaining study, I will go ahead and stratify this by study type. So over on this side, you'll see as we are putting in the plot parameters, take a second to examine the other features that we have on the left here. Once we put in our plot parameters, we are going to be able to see summary statistics and contingency tables and a plot grid and individual plots. We are going to be able to visualize this data. So here I'm going to put in type for the study type. And let's again wait a few seconds. App is taking a few seconds to load. So here I'm choosing the cluster study type. And once this loads, we will be able to go ahead and look on the left here at the results of the parameters that we just put in. All right, so those are our parameters selected. You will be able to see some summary statistics over on this side, like odds ratio and relative risk. The other thing you will be able to scroll through is contingency tables. Give it a few seconds to update. So the plot grid is going to show us a combination of all the different plots, and then the individual plots are over here. So let's go back and see if the summary statistics or the contingency table have updated. All right, let's visualize the plot grid. So you can see that as I hover over here, I'm able to see the proportions of participants that fall within that particular bar. So the blue portion of the bar is showing us the proportion of positive Schisto cases, and the green is the proportion of negative ones. And what you can see here is how the proportion of positive Schisto cases changes over the course of the study from year 1 to year 5. The upper row is the gaining control study, which was the higher Schisto villages. And the lower row here is the sustaining control studies, which were the moderate prevalence or moderate to low prevalence villages. The first bars of every pair show you our study population that we selected, the ones that the participants from Kenya that had treatment every year that were 9 to 12-year-olds. So the first bar is what we will pay attention to. So let's take a look at what these graphs are showing. What this is showing you is that in the high prevalence villages, look at the proportions. 0.65 or 65% of participants were schistopositive at baseline. And over the years of the study, this proportion went down to steadily to 58%, then to 47%, and then to 38%, and finally at about 34% by the end of the study. So this is showing you really quickly that schisto prevalence went down by half in these participants over the course of the study, and MDA was working like it should. What happens in the bottom row? Here, 18% of participants were schistopositive at the beginning, and the number went down to half, or about 8% after the first treatment, and stayed there, and ended up at 7% prevalence. So this is showing us that MDA brings down prevalence, but there is a plateau. It doesn't go down to zero, and likely other interventions will be needed to bring down the prevalence further. And this was exactly one of the conclusions from the publications that came from this study. So this search and visualization were a demonstration of the interactive features of ClinEpiDB. You can explore data, you can look for interesting patterns, you can generate hypotheses just by pointing and clicking on this website. So what if you prefer downloading the data? So let me just demonstrate starting from this table. So some studies are public. So if we choose a study that made its data completely public, if you want to download the data, you will simply get a link to the files that you can download on your computer and start analyzing with your favorite program. But if you look at a study that is protected, what happens when you want to download the data? What it will ask you to do is to register with our site and submit a data access request that then the study providers will approve, and then you will get to download your data. So that was a little demonstration of ClinEpi. And let's, at this point, go back to our PowerPoint presentation and take a look at the other. We looked at one of the SCORE studies in detail, but what I want to do is to give you a brief overview of the other 8 studies that are on here, and you can explore them on your own as you like. The second study, that's the Schisto study that's on our site, is a cohort study that was nested within the cluster randomized trial that we just explored. It asked the question, does giving MDA actually improve morbidity measures such as anemia and stunting and wasting in school-age children? They chose participants in the first year and followed the same group over the course of the study. If you look at data from this study, you will find demographics and parasitological data as for the other SCORE studies. But in addition, you will also find data on growth measures like height and weight of participants, and other data such as hemoglobin levels. The major finding of this study was that any form of MDA decreases morbidity, but that in aggregate, it is better to give MDA every year. Next, we come to the 4 haematobium studies, or the urinary form of schistosomiasis. We have already looked at one cluster randomized trial in some detail, and the 4 haematobium studies that I will present are all based on a very similar cluster randomized design. This one was done in Mozambique, and it asked the exact same questions about optimal MDA venue and timing. The major finding in this study was that when the arms are grouped together, 4 rounds of treatment, regardless of whether it was community-based or school-based, resulted in a significantly greater reduction in S haematobium prevalence than 2 rounds of treatments. That is, if it had any intermittent or consecutive holiday years included over a 5-year period. Next, in the Niger S haematobium cluster randomized trial, they compared once-a-year treatment to twice-a-year treatment in each study arm, and found that the relative reduction of prevalence was greater in biannual compared to annual treatment across all arms. However, the only significant difference was seen in areas with a high starting prevalence. The Zanzibar Cluster Randomized Trial was a pilot Schisto elimination study. It asked whether we can augment MDA with other interventions to nudge communities towards eliminating Schisto. So they compared 3 interventions: either MDA alone or MDA in conjunction with snail control, in which a chemical mollusciciding agent is added to water to kill the intermediate snail hosts. Or they used MDA with behavior change interventions, such as teaching children to play in areas or in ways that did not expose them to Schisto, or building structures such as washing platforms to give community members an alternative to washing clothes in Schisto-infested waters. And this study found that snail control or behavioral interventions did not significantly boost the effect of MDA. Finally, the seasonal transmission cluster randomized trial was done in northern and central Côte d'Ivoire, where Schisto transmission is seasonal and elimination might be achieved. Here, they tested MDA before the peak Schisto transmission season or after. A third arm tested 2 MDAs done both before and after the peak transmission season. And the last one paired MDA with snail control. And the findings from this study will be published soon. The last 3 SCORE datasets all focus on improved field assays for S. mansoni, that is, intestinal Schisto. The point of contact CCA test is a newer diagnostic test for S. mansoni compared to the classic Cato-CAT stool exam. The point of contact CCA assay is more convenient because it uses a urine sample rather than a stool sample. It is more sensitive, which makes it valuable in low prevalence areas where stool exams may miss infections, and it is field applicable and does not need a microscope. One of the SCORE datasets evaluated this new POC-CCA test in 5 different countries as an alternative to the stool exam. The finding was that one POC-CCA test was more sensitive but less specific than multiple microscopy smears from one stool, and that this new test was a suitable replacement for conventional testing. And the last 2 datasets are set in Rwanda and Burundi. Both countries were considered candidates for Schisto elimination, and they had countrywide mapping using both tests, the older Cato-CATs and the newer CCA tests. Using the more sensitive CCA tests shows that S. mansoni is still widespread in Rwanda and Burundi, although its average intensity is probably quite low. So data from all of these 9 studies are available in ClinEpi, and we welcome you to explore the data and contact us if you have any questions about it. And that concludes our tour of the schistosomiasis datasets on ClinEpiDB. Please stay tuned for more features such as a DIY workspace where users will be able to upload and visualize their own data, and a redesigned workspace with fresh new visualization tools. And with that, I thank you for your attention, and we can spend the rest of the hour answering your questions. Thank you.
So just as a reminder, please feel free to enter questions into the question box. It seems there's one that just came in. Is there going to be any new data on Schistosoma mansoni coming out soon?
We don't anticipate any new data from the SCORE studies. But data providers are welcome to contact us if they have any data sets that they would like to see in here.
Another question from earlier that I think you also answered after it was asked was who should be contacted to get access to protected data, ClinEpi or the person reported in the data set as the collector?
Okay, so to answer that, let me see if I can bring up one of these. So what I'll try to do here is to log in with my personal email ID, which is not my work ID, so I don't have access to data sets, and then see if I can show you how to access data. So here it's saying, please acquire research approval. So as you register with just your name and your email address, that's what I did right here. And then I submit a data request form, and it will pop up this sort of form. So what is already filled here is the date of request and my name and my organization. And the questions you will ask, different studies have slightly different questions, but the kind of questions you will be asked are purpose for which the data will be used, and have you discussed your request with the team? It's not necessary, but if you have, then we would just like to know it. The sort of questions you will be asking from this data, the analysis plan, and what you plan to do with the results. When you submit this, it sends an automatic email to ClinEpi as well as the study providers. And there are two different types of access. So all of the SCORE data has a passive approval process, which means that the SCORE study providers have decided to make this data public. They want you to fill this form only so that they can have a record of who has downloaded the data and what they want to do with it. So the minute you submit this request, you will get access to the data. You will get an email back saying you have access to the data and you will be free to download the data. They just want to know who you are and what you plan to do with the data for their own records. So some other studies have a level of access where your email will go to the study team and they get a few days to decide whether to approve your request. And then you will hear back from them. And if we need some more information from you about what you want to do with the data, then we will contact you for that. So it's a very straightforward process, and everything happens through the ClinEpi website. Let me show you a quick example of how your requests are. This is MALED, which is a malnutrition and enteric disease study that's on ClinEpi. So here on the study page, study request record. So this is a public table of who has asked for access to this data set, where they're from, when they asked for access, and what the purpose of their request was. So this will tell you how many people have asked for this data and what they're planning to do with it. So this is just transparency in, knowing what research questions are being asked of this data. I hope that answers your question.
That's great. Another question is there a typical or minimum size of study that you would host?
Sheena, can you answer that?
Sure. So there's a wide variety. So the SCORE studies tend to have very few variables, but have a lot of participants. I think they have 20, 30 variables loaded, but a number of participants. So that's kind of one end. MALED is kind of on the other end. It has millions of observations and also, I think, something like 1,000 variables. So there isn't really a typical study size. Sorry, I think my dog's about to.
I think more importantly, more than the study size, we look at whether this data will be valuable for secondary data analysis. Those are the sorts of things that we think about. We are just in favor of facilitating data access and secondary data use and data sharing. That's the purpose of ClinEpi. So I don't think the size of the data set matters. If you have a study that you would be interested in, chatting with us about, feel free to use this contact us link and just write to us.
Yeah. So then another question from someone is, do I need a supervisor or should I contact site PI prior to?
So again, that's a great question. And it's completely up to you whether you want to contact the PI. If it's something that you think generally, if it's something that you want to be doing a serious analysis that you want to maybe publish, we would highly encourage you to contact the site PIs, because for publication you will want to have their cooperation and their assent. But if you're just exploring the data, it may be fine to just get access and look at it. And if you think that you will want to go further with the data, then contact the PI. That would be okay too. So it all depends on probably what you want to do with the data.
Yeah, and I think that person was also asking if they should submit the request or their supervisor should submit the request. And I think either is appropriate.
Yes, completely, yes. We do anticipate a lot of students using this data, and actually we know that a lot of students use this data to just learn more about data analysis and try to replicate analyses that have been done in published papers so that they get better at data analysis, and all of those are valid uses of ClinEpi.
Yeah, and I would say if you're submitting the request, it can also be a good idea to just include your supervisor's name, anyone else who might look at this data as well. You should include that in the request, who the team is.
Another question we have is, depending on local ethical regulations, we may need provision for specific agreements for, do we need provisions for specific agreements from subjects under investigation concerning a future provision of study data to ClinEpi?
Right.
Do you have any suggestions or experience with that?
Well, I think I should have mentioned this, and let me mention it now, that all data that's available on ClinEpi is de-identified. There is no way to link particular individuals to any of the data that's available here, even though it's on a participant level. We also go ahead and obfuscate dates, like dates of birth and dates of visit. We obfuscate those dates by a week so that you do not have the exact birth date of a particular individual. So just wanted to mention that. And the question about submitting data to us, or can you repeat the question, Steph?
Sure. The question is, depending on local ethical regulations, we may need to get provisions of specific agreements from the subjects under investigation.
Yeah, so we have some in our frequently asked questions section under the About menu. We do have a little bit of information that touches on this. We do recommend that people clear this with their IRB or their equivalent of an IRB, depending on what country you're in, and basically get ethical clearance to share data on something like ClinEpiDB. That link, that IRB data share and guidance document, includes some information that you may want to consider when constructing your consent forms, and that you may want to consider when talking to your ethical committee about sharing the data with ClinEpiDB.
Okay,
and then another.
Oh, you got it?
Go ahead, Steph.
Another question we have is, after a new analysis, can someone refer to the output and acknowledge the source of it if it's not in the publication?
Yes, I think so. You are definitely, when you do an analysis, you can save the analysis, you can copy the graph, and all the acknowledgement questions really come up if you're publishing it. But if you were to say present this data and so on, and just acknowledge that it was done using this study on ClinEpi, I think that should be fine, if I understand the question correctly. Sheena, do you have any thoughts?
Yeah, same as you. And then just to point out that there's an FAQ just above that says, how do I cite ClinEpiDB? And that includes how to cite a data set. So if you use data from ClinEpiDB, how to cite that specific data. So you're actually citing the people who generated the data. And then if it's always a good idea also, if there are publications on that data set, to cite whichever of those are relevant when to your analysis.
Mm-hmm.
We have another question. Is there any data on schistoproteomics on this platform? And if there isn't, could you recommend one?
You know, I guess I don't know the answer to that. Sheena, do you know if there is any Schisto data on any of the genomic sites? That's something that we may be able to find out for you.
No. So we used to have SchistoDB, but it's no longer supported. I think Wormbase or some other site like that includes supports schistosomiasis data, but I'm not positive.
If you like, to the person asking this question, feel free to email us and we will find out for you. But I'm sorry, I don't know the exact answer.
Another question, if someone wants to work with two different data sets simultaneously, for example, MALI-D and GEMS, to explore a single outcome, is that possible? And if yes, how do I request it?
So that's a great question. And that is a functionality that we are developing at this minute. So right now, we do not have the functionality to query two different data sets at the same time, but it is something, the cross data set analysis is something we are working on. Sheena, do you have anything to add?
This
so
also may be
I can. Yeah, so I would recommend that if someone wants to work with both MALI-D and GEMS data to explore a single outcome, the best way to do this would be to request access to each of those studies. So you would submit two separate data access requests to ClinEpi, and then you can go ahead and download both of those data sets, and you can perform your analyses on your own computer, but not on the website.
So one thing I should also point out is that this is something that you don't see on the site because it's happening behind the scenes, is that in ClinEpi, we are working very hard to harmonize the ontology across data sets. So for instance, a label means the exact same thing across data sets. So when we call something a fever, it has the same definition across data sets. We work behind the scenes to harmonize that. And that makes it actually easier to work with data sets together. Even if you can't do it on the website at the moment, you can still know that the same label means the same thing in two different data sets.
Someone commented that schistosomiasis data is included on Wormbase, so that may be a place for that other person to look.
That's great to know.
And then the same person was asking if they can have our email, and our email address is help@clinepidb.org. And you can also use the contact us link. It sends it to the same email address.
Sheena, would you put that in the chat maybe?
Yeah.
The email address. Yeah, but definitely feel free to just, if you go on the main page, use the contact us link. We are very responsive, so you will get a response.
It looks like that's all of the questions that have been asked.
Well, thank you everybody for your time. We really appreciate you coming out and hearing our webinar. And like we said, please, this is a site that you should play with and explore on your own, and contact us if we can help in any way. Thank you for being here and enjoy the rest of your day. Bye-bye.